Hello everyone. This video is in continuation with my previous video where I had explained you about mono-hybrid cross from the chapter Genetics, Mendel's experiments on pea plant. Now today I am explaining you dihybrid cross. First of all, what is dihybrid cross? A cross between two individuals, they may be plants. So cross between two individuals with two characters with their contrasting traits is known as a dihybrid cross. What did Mendel do in this experiment? He considered two characters on pea plant. As I had already told you previously that pea plant has several characters with contrasting traits. He selected two pea plants, one which was producing round and yellow seeds. Now see, round is the shape of seed and yellow is the color of seed. So these are two characters, round and yellow. Now it has means another character was wrinkled with green seeds. So round and wrinkled, these are two contrasting traits, and yellow and green are two contrasting traits for the same character. So he has taken the shape of the seed, round or wrinkled, and he took color of the seed, that was yellow and green. When he crossed these two plants, see, this is phenotype. I have written in short. Here it is for parents. Parents were with round yellow seed and wrinkled green seed. So this is phenotype. I had already told you phenotype means the external appearance. So phenotype is round yellow and wrinkled green. Genotype is genetic constitution. Each character will be controlled by two genes or two factors. Round is controlled by RR, capital RR, and yellow is controlled by YY, capital YY. Since this is dominant character, we will denote it with capital letters. This side wrinkled RR, here it was capital R. So we will be using two small r for one character and two small y for green character. This is a recessive trait, so we will denote it with small letters. One character controlled by two factors, so two characters will be controlled by four factors. These gametes are produced during sexual reproduction. Gametes are produced. So here two types of gametes are produced, RY and RY, small r and small y. In F1 generation, when these gametes fused together to form the progeny, you can see these are hybrids. Now two more terms can be explained from here. One is homozygous and the other is heterozygous. Homozygous is this condition when two factors are same. Two factors of an individual are same for one character. R homozygous, Y homozygous. Here R and R. If we take this, this will be denoting round. So this is heterozygous because both the factors controlling this character round are not similar, they are different. And capital Y and small y is also heterozygous condition. Now this is how to read this. Now if you have to read, take two factors together. So R will be round and Y and small y will be yellow. So in first progeny, that means first generation, all the plants he found, they were round and with yellow seeds. Now what happened in second generation? Now this is result of second generation. This is a Punnett square. Even in monohybrid cross you can show the result with using Punnett square. What is Punnett square? In this Punnett square, these gametes which are produced in second generation are placed on one side of the square, and the gametes of female, this can be gametes of male or gametes of females, and gametes of male or female can be placed on other side of the square. Now when we cross, now what type of gametes are formed? This also you can see easily. how to write, because if you write it wrong, the whole combination will be wrong. So this is very, very important. 4 types of gametes will be formed from this progeny of first generation. So in second generation, when this progeny means new offspring were self-pollinated, 4 types of gametes were produced. Capital R, capital Y, capital R, small r, capital Y, and small r, small y. Same gametes you can place this side also. See, these are same gametes placed to this side. When we cross, how to cross them? Take first gamete, cross with all 4. Take this first, cross with first. So R, Y, Y. Second is R, Y, small y. R, capital Y, R, that is capital R, small r, capital Y and small y. And in this way you can cross all gametes till last square. Take this second and cross with all to see the result here one by one. Similarly, take this gamete RY, capital Y, and cross with all to fill this line. Then last small r, small y, cross with all. You will be getting these results. Now, how to write the phenotype? This is all genotype. How to write phenotype? For writing phenotype, take 2 factors together. R, round. Capital Y, yellow. If both are capital, it will be showing the dominant trait. If both are capital here, it will be showing the dominant trait. But if one is capital, in this combination you can see R is round, capital Y and small y. Only one y is there, but it is sufficient to express itself. So it will be round and yellow. Here again, both are heterozygous, but it will be showing the dominant trait. So round, yellow. In this way you can read round, yellow. This is round, but it is green. Both are small. So recessive trait is expressed only when both are small. That is recessive. Similarly, if both are small r, as you can see here, it will represent wrinkle. Both are small, it will represent wrinkle. Both are small, it will represent green. So out of these all 16 squares, if the result in F2 generation was seen, it was found that from here only that round yellow seeds were 9, round green were 3, wrinkled yellow were 3, and wrinkled green was only 1. That means this recessive trait, which was hidden in first generation, reappeared in second generation, F2 generation, but only 1. So the ratio of phenotypic ratio of F2 generation of dihybrid cross is 9:3:3:1. And genotypic ratio, since it is very complicated, very complex, so you don't write this ratio. And in answer also you will be writing that genotype ratio is very complex. So this is all about dihybrid cross. In my next video, I will be explaining you with some important terms.